All right, so this is our primary mission. We provide academic researchers with the tools and expertise to enable high-impact, large-scale biology and drug discovery in a state-of-the-art open access facility. So to achieve that, we have a suite of automated liquid handling instruments and detection instruments, plus the staff to be able know how to use those instruments and all the ins and outs of high throughput screening. So as secondary activities to enable the drug screening, we also curate libraries of drug-like small molecules and FDA-approved drugs. And because the instruments are not booked full-time by all the high throughput work that we do, we also make them available to walk-up users that can get trained on the use of them, and both the liquid handlers and the detection instruments. So on this side, I tried to kind of list everything I could think of that we support. And they fall broadly into three categories: the early steps in drug discovery, large-scale basic biology projects, and microplate assays in general. There's a lot listed here, but I think the key takeaway is that for the most part, if your assay is compatible with microplates, we can, get them working on our instruments and optimize them for you. So before I get into what we do, I wanted to cover the question of why should you consider using the high throughput technologies that we offer. So, of course, it can drastically decrease the time it takes to complete an assay, probing a large number of conditions in parallel. And it can also decrease the cost per condition. So in most cases, we're miniaturizing the assays that we're running, and so you end up using a lot less reagent, which can save you quite a bit of money. And then, and those, both that time reduction and cost reduction can lead to an increase in scope, just because you have the time and money to do it. And then also because we're using these automated liquid handlers, it often will improve your data quality and reproducibility. So now I'm going to talk about our resources. So for me, our most valuable asset is our staff and all the years of experience that we all can offer to you. So collectively, we have 60 years of screening and drug discovery experience. There's 5 of us on the team, and each of us brings our own kind of set of unique skills and experience to the team. All right, so I'm going to just really fast go through all our instruments that we make available to our users. If anybody has any specific questions about what instruments are good for your particular project, you can reach out to me offline or after everything is over. So here's a couple of bulk dispensers that we have: the Thermo Combi and the Integra Viafill. Bulk dispensers are just instruments that are used to quickly dispense larger volumes of kind of the same reagent to all the wells on a plate, or most of the wells on the plate on a microplate. And for our automated liquid handlers, these use the basic positive displacement that everybody's sort of familiar with pipetters. We have a Tecan Fluent and an Agilent Bravo. So both of these have decks where they can hold a number of plates for complicated workflows. And the Fluent also has a vacuum manifold. It can do tube to plate rearraying, and it has a bunch of other peripheral devices on the deck for, you know, for like complete walk-away automation for different assays. So we do have a couple of unique dispensers. The Formulatrix Mantis uses a microfluidic chip for dispensing liquids. It's more like it's similar, I think, to the bulk dispensers, but it does small volumes. And then the Mosquito uses disposable piston-based tips. And they each have kind of their niche use that we use them for. And the Mosquito in particular is popular with the crystallographers. And also we've optimized a miniaturized qPCR workflow on the Mosquito. We have an instrument called the LabSite Echo 555. Although LabSite was purchased by Beckman, so I'm not sure what they're calling it now. But the Echo is an acoustic liquid handler, which means that it generates these sound waves that eject precisely sized 2.5 nanoliter droplets from a source plate to a destination plate. And then it can pretty much dispense any volume as long as it's a multiple of that 2.5 nanoliters. And it can go from any well on the source plate into any well on the destination plate. And it's housed inside a robotic platform when we're doing kind of a lot of plates going through a big workflow. And then finally, for plate washing, we have a Tecan HydroSpeed, which is optimized for washing adherent cells in microplates. All right, so moving on to the detection instruments. We have a couple of multi-mode plate readers. They can do absorbance, fluorescence, fluorescent polarization, time resolve, fluorescence, luminescence, alpha screen, fret. So just pretty much anything that you do in a microplate. And then each of them kind of have their some unique attributes that, so one of the Tecan has stackers that we use for the high throughput screening, and then the Biotech has a reagent injector for other types of assays that need, you know, specifically timed injections of the reagent. So you might not associate it with high throughput screening, but we also have a qPCR instrument that's set up for 384 well plates. We use it for the miniaturized qPCR reactions, and also we do use it for something called fluorescent thermal shift assays. It's used in screens for protein binders, and I'll talk about that assay a little bit more later. And then finally, we have a Molecular Devices Image Express Micro Confocal, high content imager. So it's integrated with a robotic arm and an automated incubator, and soon will be integrated with the Bravo as well. So, we'll have complete automated imaging pipeline. And then we're also working with Dr. Julie Kim from Feinberg on a microfluidics device that will integrate into the system, and it can be used for multi-tissue organite screening. Oh, and then finally, sorry, that was not the last one. We just got a new WaveFront Biosciences Panoptic. It is a kinetic imaging plate reader, and I'm not going to talk about it because Dave Weaver, the president of WaveFront Biosciences, is going to be talking after me and telling you guys about how everything that Panoptic can do. right, so in the next few slides, I'm going to be talking about a few examples of the different assays we've used in screens. In the interest of time, it's not a comprehensive list, so I just wanted to throw this slide out there to give you a pretty good idea of all the different assays we can accommodate. So for on the biochemical side, you can we do both binding and functional assays. For example, protein-protein, protein-ligand, protein DNA assays. We can do different enzymatic assays with kinases, deacetylases, proteases, ligases, you know, the list goes on. And then for cellular assays, they can be either targeted or phenotypic. So for examples, on the targeted side, we've done GPCR screens, ion channel, signaling, reporter genes. And then on the phenotypic side, we've done cell death, morphology, effector secretion, you know, sort of, you know, anything that you can think of that we can do in a microplate, we probably can do it. Okay, so moving into the specific examples, I mentioned the fluorescent thermal shift previously. FTS uses an environmentally sensitive dye that quantifies the melting of proteins in real time. So compounds or ligands that produce a significant shift in the melting temperature of the target protein are identified as putative binders. So we've used FTS for drug screening, ligand ID, crystallography, optimization, characterizing mutant proteins, you know, among other things. So since FTS is a pretty cheap assay, provided the protein has a pretty well-defined melting temperature, a melting profile rather, sorry, we can use it for large-scale screening. And so here's an example of a data set for a screen of 50,000 compounds. That works, yay. And so green, the green indicates, sorry, this is hard to do. The compounds that yielded a shift of 2 degrees or more. And so by using that 384 qPCR machine, we can screen through thousands of compounds per day. So another assay we used quite a bit is called Alpha Screen or Alpha ELISA. So that's it's made by PerkinElmer, and it's a homogeneous no wash replacement for ELISA screen. And Alpha stands for Amplified Luminescent Proximity Homogeneous Assay, sort of a mouthful, but the fundamental principle of the technology relies on the binding of 2 different molecules. Not very good at this. I'm going to skip the pointer. So it relies on the binding of the 2 different molecules to the 2 different alpha screen beads. And then when there's an interaction between the 2 molecules, it will bring those beads together. And once they're in proximity to each other, there's an energy transfer from the donor to the acceptor bead. And that transfer results in a chemiluminescent signal. So we use the alpha technology in assays to assess biomolecular interactions or to measure the formation or depletion of analytes, specific analytes. So it can be used in both biochemical and cellular assays, and the fundamental aspect of it works pretty much exactly the same, whether it's in cells or in cell-free solution. So fluorescent polarization, or FP, is based on the measurement of a molecule's rotation in solution. And so we use it to study the molecular interactions in solution. We found it to be a pretty robust assay for screening or for inhibitors of protein-protein interactions and other molecular interactions. Okay, so a number of companies, but in particular for us, we use Promega. They've harnessed that luminescent technologies in a really diverse array of kits that to measure anything from cell proliferation, enzymatic reactions, or gene expression. We go through the cell titer glow reagent from Promega, almost like water. So we do a lot of proliferation and toxicity assays. We also all use luciferase as a reporter protein. So when we're screening for modulators of gene expression. All right, so Promega has also developed a really neat kit to measure protein-protein interactions called nanobit. So it can measure the interactions within cells, and the assay requires a little bit of cell engineering, but once the cells are generated, it's a pretty powerful technology for measuring protein interactions in live cells. Right, another assay we've optimized is, we call it in-cell Western assay. I've also seen it called cytoblots, cell-based ELISA, in-cell ELISA, or fast-activated cell-based ELISA. So it allows us to measure things such as protein phosphorylation, GPCR activation, protein expression, viral titer, the activity of PROTACs. It's pretty versatile, and so we're measuring those things directly in fixed cells without the gels or the blots in Western blotting, and so it is a high throughput compatible version of Western blotting. And then I mentioned our Image Express high content imager previously. So with the use of automated fluorescent microscope, we can measure readouts such as cell morphology or protein localization, readouts that are not measurable on the plate readers or even by flow cytometry. So one of the ones that we do pretty regularly is neurite outgrowth measurements. So the software can measure the number of cells, the length of the neurites, thickness, intensity, branch points. There's quite a few different outputs that it can measure. And another one we use quite a bit is for 3D spheroid assays. So we've done both screens where we use the transmitted light and no probes to visualize and measure the spheroids. And then also we can use dyes such as calcine AM and FD to get the live and dead cells in the spheroids. All right, so for this slide, I just wanted to throw up, this is a resource for everyone. This is just a list of the different compound libraries we have available for screening at the core. We have about 6,000 of the compounds with known activity, so they're annotated. They include the FDA library in the clinical collection. In addition, we have about 40,000 compounds in our focused sets. And then we have about over 100,000 in the diversity sets. So these were purchased already dissolved in DMSO at 10 millimolar, and they are arrayed in microplates. So they're really only for the high throughput screening. We can't distribute them individually in any quantity. Okay, so with that, I'm going to turn it over to Dave. He's the president of WaveFront Biosciences. He's an expert in ion channel screening and high throughput screening. And so when the commercially available instruments that were available didn't do what he wanted them to do, he went and built one for himself. So we have one of his instruments here at Northwestern, and it's called the Panoptic. And so bear with us for a minute while we get Dave set up to present. Right? Yeah.
Great. All right. Thanks very much, Sara. Sara said, my name's Dave Weaver, WaveFront Biosciences. I've had a few different careers over the years. First at Bristol Myers Squibb, where I became familiar with this type of plate reader. And then for the last 20 years at Vanderbilt University, where we developed the first WaveFront Panoptic. So first, just what is a kinetic imaging plate reader? I'm going to try to put on the laser. Well, I tried and already messed up. Let's see. All right, so what is a kinetic imaging plate reader? Can you all see my red dot? I can't hear anybody talking. I'm hoping somebody can hear me. Let me...
You see the pointer.
Yes, Dave.
Yeah, okay, great. Okay, great. Okay, so if this is a typical epifluorescence microscope where you've got some type of excitation, in this case, you're shooting it onto a dichroic beam splitter onto the sample, exciting some fluor, then the resultant fluorescence coming back, going through an emission filter into a detector. And if this is, in this case, an upright fluorescence microscope, a kinetic imaging plate reader, sometimes I refer to it as a macroscope because the guts of the optics look very similar. Excitation source, filter, sometimes has a dichroic beam splitter, sometimes it doesn't, emission filter in a camera. But instead of focusing the light on a small sample, what a kinetic imaging plate reader does is spread the light over an entire microplate. So it is anything that fits in an SBS standard size microplate from 6 wells to 1,536 wells can fit in an instrument like this. And then the other part of it that's important is a pipetter head, so a 96 or 384 well pipetter, because an instrument like this, what it's best suited for is taking low spatial resolution measurements, high temporal resolution measurements all at the same time. So what I'm going to hope to show you is if this little movie will run, sure that it will. Well, okay. So instead of me futzing with it, I'm just going to say that it's not going to go. So this is what the image looks like. So you can see it's a low spatial resolution image. Each well is just represented by a glowing dot. These glowing dots could be cells either in suspension or adhered to the bottom of the plate, or this could be a cell-free assay. As long as whatever is in here is either fluorescent or luminescent, you'll be able to generate a signal. And this is what the data looks like. So if you're taking pictures of this, we can go up to 100 frames per second. Most people typically are running at about a frame per second. So you're taking a picture every second, and what you end up with is a movie. You can then take the pixel values for each one of these wells and express them on the y-axis versus time, and what you end up with is a wave. So you're taking a low spatial resolution, high temporal resolution movie and turning it into a wave, and this waveform then represents something interesting from a biological perspective, hopefully. And using something like this, you can go from maybe 30 kinetic assays a day on a microscope to 30,000 or more assays a day on something like a kinetic imaging plate reader. So the WaveFront Panoptic is a specific example. Sometimes I refer to it as a laboratory in a box, because not only does it have this detection modality, it also has a fully functional adjuvant Bravo. So Sara mentioned the adjuvant Bravo is one of the liquid handling modalities found in y'all's high throughput assay facility. So this is the exact same instrument. And it can do transfers 96, 384, and 384 to 1536. It has 9 deck positions. So you can actually design very flexible and complex experiments because it utilizes Agilent's vWorks robotic scheduling system. And because it has a gripper, it's hard to see on here, you can do things like lidding and delidding plates, shuffling plates around the deck. So once you have your assay loaded up in here, you can close the box, control the temperature ambient to 40 degrees, and walk away while your either cell-based or cell-free kinetic functional assay is running. The WaveFront Panoptic uses a solid-state laser source. So these are discrete 1-watt solid-state lasers. These are the wavelengths, so they span most of the visible spectrum, and they're compatible with the most commonly used fluors, particularly the fluorescein-based fluors that excite around 490 nanometers, but also a variety of other ones like the Psi3 related fluors, some of the longer wavelength fluorescent proteins, and some shorter wavelength coumarin-derived fluors. Because these are discrete light sources, it's very amenable to multiplexing. So this is this wavelength center frequency plus or minus about 2 nanometers. So it's easy to resolve different fluors at the same time. And because there's fast switching, you can switch back and forth from one excitation to another to do things like enable photopharmacological and optogenetic manipulation of your sample while you're at the same time making this high-speed movie of changes in fluorescence or luminescent signals. So this type of plate reader was really designed to do cell-based functional assays. So the first one was made by a company called Noveltech in about 1994. And over the course of the last 20 or so years now, I guess 30, which is making me feel old, have been mostly used for these cell-based calcium-sensitive fluorescent indicator assays, predominantly in the G-protein coupled receptor space in drug discovery, but also in ion channel and other types of biology. And the way things like this work is whether you've got a 7-transmembrane receptor, aka GPCR, a plasma membrane calcium channel, or an intracellular calcium channel, you can load cells with fluorescent small molecule calcium indicators, or these days also genetically encoded calcium indicators. And as calcium increases inside the cell, using the kinetic imaging plate reader, you can get data that look like this. So in this case, I've removed extracellular calcium. So I'm looking at only the calcium that's coming through this IP3 receptor that's triggered by activation of the GPCR, activation of phospholipase C, cleavage of PIP2, generation of IP3, and an increase in intracellular calcium. And then if I add calcium back to the outside of the cell, I can see calcium coming through, in this case, a TRP channel, TRPC6 on these cells. So you can see you can get very nice concentration-dependent activity and resolve intracellular from extracellular calcium contributions to your assay. So this is the kind of thing that I did when I was a postdoc. And when I saw my first kinetic imaging plate reader, I could tell that I could do pretty much every assay that I did in a 3.5-year postdoc in one afternoon. So that's an example of the kind of power that you can get from an instrument like this. So you can use these instruments to do all different kinds of screens. Here I'm giving an example of one that's commonly used in the GPCR field these days. It's sometimes referred to as a triple addition assay, used to detect positive allosteric modulators. It doesn't have to be a cell-based assay, and it doesn't have to be a GPCR, but this just kind of exemplifies what one might do. In an experiment like this, the first thing we do is add a test compound and ask, does the test compound do anything to intracellular calcium on its own? And in this case, the red is the highest concentration of the test compound, and the cooler colors are lower concentration. So you can see at the very highest concentration, the test compound is causing a change in intracellular calcium. When we add an EC20 concentration of the agonist to this GPCR, so this is a muscarinic acetylcholine receptor, M3, and a hex cell. So if I add an EC20 concentration of the agonist, I'm asking the question, does the presence of this other test compound amplify the signal? And you can see that it does. So at a maximally effective concentration of this compound, it dramatically increases the efficacy of acetylcholine. And in this third edition, I can ask a question that's typically used to see if this molecule is an inhibitor of the response. So in this case, I'm adding an EC80 concentration of acetylcholine and looking for an effect there. So as you can see, in terms of liquid handling moves, quite a complex assay, but a lot of information is generated in a short amount of time. So this is a short assay where you generate a lot of information, and you're doing this in 384 wells in parallel. As I mentioned, by using these discrete, rapidly switching light sources, you can do multiplexed assays, which is to say you're doing more than one assay in the same well at the same time. And I'm going to give you an example here of a modality that's looking at mitochondrial membrane potential. So as I'm sure you know, the mitochondria generates a large membrane potential. And as such, you can use a fluorescent indicator like this. I just started introducing the next slide. So forget about the last 30 seconds of what I said. I intended to be talking about mitochondrial calcium. I'll talk about mitochondrial membrane potential in a moment. So in this case with multiplexing, I'm looking at two different calcium-sensitive fluorescent dyes. Flow 8 is a cousin of Flow 4. When you load it into cells, it's typically found mostly in the cytoplasm. So you can use Flow 8 to load a calcium indicator into the cytoplasm. You can use a cousin of Flow 8 that's based on a rhodamine that tends to accumulate in polarized mitochondria. So mitochondria that are maintaining their normal membrane potential, being negative on the inside, they tend to accumulate this rhodamine-based dye. So what you end up with is cells where their mitochondria are predominantly loaded with a red calcium indicator, the cytoplasm predominantly loaded with a green calcium indicator. And by using fast switching, you can generate both of these sets of data at the same time, where the green signal represents a change in intracellular calcium, primarily in the cytoplasm, where the red signal reflects the change in intracellular calcium inside the mitochondria. Now, if you remember what I was saying a few seconds ago, this is a case where you can utilize fast switching to interrogate a dual excitation, dual emission sensor. So JC10 and its cousin JC1 are fluorescent dyes that when they experience membrane potential inside the mitochondrial matrix, they form these what are referred to as J aggregates that have a red-shifted excitation and emission spectrum compared to the JC10 monomers that predominate both in the cytoplasm and the mitochondrial matrix when the mitochondria is depolarized. So you can use these as a cell-based functional assay to ask questions about mitochondrial health and whether the mitochondria is able to maintain its negative membrane potential. So this is an example of what data look like here. Using fast switching, we can interrogate the JC10 monomer, and the monomer fluorescence increases as I add a mitochondrial poison, CCCP, and the aggregate fluorescence decreases simultaneously. So you can get both of these signals at the same time. This is useful for a wide variety of ratiometric indicators and other things like Forrester resonance energy transfer assays, where you're looking at a dipole-dipole interaction that conveys excitation energy from one fluor to another. And finally, the camera that's used in the Panoptic is a very sensitive camera that can be used for ultra-low light assays. In this case, what I'm showing is a luciferase-based assay where a cyclic AMP binding moiety has been engineered into a firefly luciferase, such that when cyclic AMP binds, the luciferase assay is reconstituted. So now you get a degree of luminescence that's proportional to the increase in intracellular cyclic AMP. As many of you know, there are many ways to measure intracellular cyclic AMP, but most all of them are single endpoint assays that rely on you to have to understand the cyclic AMP kinetics inside your cell to get an answer, and often also to inhibit the cyclic AMP phosphodiesterase. So in this case, you can actually measure changes in cyclic nucleotide dynamics in real time. In this case, we have the melanocortin-4 receptor expressed in HEC cells. We add the agonist ligand alpha MSH, and we can see a nice concentration-dependent increase in luminescence. So this can be used for a variety of cell-based and cell-free luciferase assays, and a cousin of FRET, this bioluminescence resonance energy transfer. So you can read this in things like the variety of nanoluke assays, and also recently the nanobret assays, which take a halotag, a nanoluciferase, and some newly developed synthetic calcium indicators to enable high throughput bioluminescence-based calcium flux assays that are becoming more and more popular, and are soon to be commercialized by Promega. Finally, a plug for another company that I'm involved with, whose reagents are ideally suited to Panoptic, and in particular ones that might be interesting to you all. This Mitavolt kit utilizes this dual excitation, dual emission dye for asking questions about mitochondrial health. A new generation of calcium indicators that have redshifted fluorescence properties that are perfectly tuned for the green laser of the Panoptic, making them GFP compatible. So you can look at GFP and this calcium gold or thallium gold assay in the same cells at the same time. And finally, an assay technology that's starting to gain some ground for people that are interested in measuring the activity of sodium-dependent transporters. So these are things like glucose transporters, amino acid transporters like glutamine transporter, and a variety of neurotransmitter transporters as well. So this is very well suited for real-time assays on an instrument like Panoptic. So that's all I've got today in terms of slides. This is me. This is how you can get in contact with me. So if you are thinking to yourself, does any of the things that I'm working on map onto a workflow that would be good for Panoptic, low spatial resolution, high temporal resolution? Of course, contact Sara first, but I'm always here for you if you've got any questions. Thanks very much.
Thanks, Dave.
Sure.
Does anybody have any questions? Or I can also just stick around afterwards for a little while if you want to kind of just get my contact info and talk to me later as well.
This is the one. I have a question.
Yes, go ahead.
So I'm interested in your that dual excitation, dual emission sensor use a JSN10, and the Y is monomer, the aggregators. What kind of mitochondrial function you can do those, and you just put one dye there and give a different drug treatment. You can separate there, and the most of the time the cells you maybe have aggregates, you have a monomer. How you separate them? Use that. That's your resolution.
Yeah. So on a modality like this, you can separate them temporally. In other words, you can watch. So mitochondria, normal healthy cells are going to be mostly red. And if you were to use something like the image express microconfocal that Sara showed, what you would see is cells with very bright red puncta that represent the inside of the mitochondria. If you depolarize the mitochondria, you'll see the mitochondria turning green and the cytoplasm becoming green. And so people often use this as one of the ways that you can indicate whether or not either the cell is healthy or the mitochondria is healthy. So, in this case, I'm using a simple kind of mitochondrial uncoupler to generate the point. But anything that might cause the mitochondria to become dysfunctional and unable to maintain its membrane potential would cause a signal that's like this. So for people that are interested in asking questions, maybe about whether or not a compound that's cytotoxic is acting through effects on the mitochondria, as an example. Maybe you're interested in looking at the switches in metabolism from, say, mitochondrial to non-mitochondrial metabolism. Assays like this could be useful for that. And they could very well be used in combination with something like the Image Express Micro, where with Image Express, you're asking questions in high spatial resolution. And in an instrument like this, you're asking questions about, what's happening in real time. How fast does a compound cause mitochondrial dysfunction, for instance?
Is that data analysis easy? When you get an image, data analysis is easy?
Yeah, so the way this works is if you look at the multi-well plate, each one of these wells, we use a digital mask, and we say the light coming from this XY coordinate on the plate is from A1, for instance. And we just express that as a fluorescence or luminescence intensity value. And then to analyze the data, it really depends on what feature of the data that you're interested in. So you might be interested in the peak. You might be interested in some decay constant. You might be interested in the area under the curve. And the software will allow you to extract values from all of those. You can place cursors at different time points and ask the question, what is the luminescence or fluorescence intensity over that time? You can express it as a variety of different things, including time constants, maxima, minima, etc. So then you have taken an image and turned it into a wave, and then extracted individual values out of that wave that you might use to generate concentration response relationships, for instance, to get potency and efficacy values. So the software will allow you to go from here to single or multiple numbers per wave easily, and then curve fitting can be accomplished on whatever your favorite curve fitting software is, whether it's GraphPad Prism or IDBS Excel Fit or whatever you typically use to generate potency and efficacy values.
All right, thank you.
Sure.
Is there any other online questions or nope? Okay. Well, thank you all so much. And I'll hopefully hear from you guys at some point.
Thanks very much. Thanks, Sarah. Thanks, everyone.